Hello everyone and welcome to Ovarian Cancer Australia's Information and Support webinar. My name is Vanessa Alford. I'm part of the support team at OCA. I'd like to begin by acknowledging the traditional owners of the land that we're on and pay my respects to elders past, present and emerging. This webinar will focus on the latest developments in ovarian cancer treatment and research. We have three presenters who are going to share some valuable information with you. We're extremely grateful for the funding received for this webinar from AstraZeneca. The webinar is interactive, so over the next hour, you can ask questions to the presenters by posting a question in the Q&A chat box. Questions will be answered at the end of each speaker's presentation. Please note that presenters cannot address individual medical cases. If you have a particular question, please call the Ovarian Cancer Australia Information and Support line on 1300 660 334. I'm extremely privileged to introduce our presenters to you. Our first presenter is Professor Clare Scott, Joint Division Head of Clinical Translation and a Laboratory Head at the Walter and Eliza Hall Institute of Medical Research, and a Medical Oncologist at Peter MacCallum Cancer Centre, Royal Melbourne and Royal Women's Hospitals. Professor Scott will summarise what we know so far regarding HRD and why it matters to women. She will discuss what some of the options may be for Australia in the coming years. Our second speaker is Associate Professor Yoland Antill, a medical oncologist and cancer genetics specialist in Victoria with appointments at Cabrini, Monash Health and Frankston and Royal Melbourne Hospitals. Associate Professor Antill will provide an update on clinical treatments for ovarian cancer and give an overview on what we can look out for in the future. We will then hear from Rhonda Chapman, who was diagnosed with stage 3 high-grade serous ovarian cancer in September 2020. Rhonda has been on several clinical trials since her diagnosis and will share her experience of being involved in these trials. So I'd like to now hand over to the microphone, Professor Clare Scott. Thank you, Clare.
Thank you, Vanessa. Just doing the important handover. Hopefully you can all see my slides now in presenter mode. So it's a great privilege to be able to give this talk to you tonight. And as Vanessa said, I'm going to concentrate on HRD. What is it? Where we're going with it and why particularly it matters to women with ovarian cancer. It is a lot to cover in 15 minutes, so I'm going to whiz through some of the concepts, but I've provided a lot of detail on the slides. These are my speaker disclosures, and they do include research support from companies who make PARP and cell PARP inhibitors, such as AstraZeneca, and but I don't accept any honoraria for any advisory boards or presentations. So tonight I'm going to divide my talk up into 3 parts. So we're going to talk about biomarkers, what they are and how they assist us with clinical trials and with studying treatments. Then an overview of HRD itself and the tests and the challenges that those tests represent. And then some practical considerations based on where we are for HRD testing in clinical practice. So firstly, just to think about biomarkers. So they have horrible names: prognostic, predictive and exploratory. So we're just going to give some examples of each of those in ovarian cancer, so then we can see a bit of a difference. So if we think about BRCA1 and 2, and these are, initially discovered in the germline, but now we know that we can think of them in the tumour as well. So they provide prognostic information, and that means how a woman is going to do in terms of her cancer overall. We know that a woman with a BRCA1 or 2 mutation is probably going to have a cancer that will do slightly better than someone without a BRCA1 or 2 mutation, particularly for BRCA2. But we also know they're predictive of response, and that is to things like carboplatin chemotherapy and to the PARP inhibitor. So both BRCA germline and somatic are predictive. Now this HRD now has gotten to the point where we do believe it's a predictive biomarker, and I'll show you the guidelines for that. And then just as an example, is MSI or MMR relates to immunotherapy, and there are certain conditions where that is also a biomarker for response to immunotherapy for certain ovarian cancers. Then exploratory biomarkers include some of these other things listed here, but they're much less well understood, which is why they're exploratory and they haven't got to the stage of being predictive yet. So we're often these days using next generation sequencing here at diagnosis, and we would like to be doing this a lot more to stratify patients so that we know up front what sort of treatments they should have. But at the moment, this is not being done in the first line, apart from for BRCA1 and 2 in the tumour, and for a number of BRCA1 and 2 related genes in the germline. Why do we care about HRD at all, and what is it? So the HR part I'm going to describe first. So that stands for homologous recombination, which is just a really long way of describing a certain type of DNA repair. So all of our cells undergo damage all the time. And the reason why most people don't get more than one cancer in their lifetime, if they're going to get a cancer, is because our cells have evolved really sophisticated ways of repairing all types of DNA. So we have multiple different types of DNA repair pathways as well. But this HR pathway is really one of, or if not the most important, because it really has high fidelity. It does a very, very good job. It takes care of both strands. It's highly accurate. And when that is abnormal, so when certain genes from that pathway are missing, we call the pathway defective, and that's the D in HRD, so HR defective or an HR defect. And what we know about a PARP inhibitor is that it also plays into this type of repair. And if you get a double whammy or a double hit, you get a cell that already has a defect in homologous recombination, and you come in with a PARP inhibitor, which affects a number of different types of DNA repair in cells and in cancer cells. You end up getting cell death. Now, the good thing about this is if you've got a cell that is normal for HR repair, like other normal cells in the body, even in women who've inherited a BRCA1 or 2 mutation, the PARP inhibitor doesn't have the same effect, and cells survive and really don't give a hoot about the PARP inhibitor. So that's very good. It gives us what we call a therapeutic window, which means there's a much better chance that we can deliver drugs like PARP inhibitors to women without causing much in the way of side effects. So this whole concept that you need 2 hits is what is called synthetic lethality. So neither hit on its own is lethal, but both hits are. So we have some ESMO recommendations published in the Annals of Oncology, and I'll go through these in a moment, but this was the first time we really looked at the predictive capability of HRD for PARP inhibitors. So this again shows you what I was just describing in another kind of a pathway. So you've got the double strand break up the top, and all of these guys come in and repair. So these are proteins found in most cells in our body. And if everything's fine, HR proficient, you get this error-free DNA repair. But if there is a problem with one of the genes, particularly with BRCA1 or 2, or other genes, so either a mutation or silencing called methylation, you end up with a functional HR deficiency. Now, these next 2 slides are complicated, but I've just put them in case you want to see some of what we then talk about, particularly with the Myriad My Choice assay. So I'm highlighting that because at the moment that is the commercial test that most people choose to buy, to pay for if they're going to have testing. And it is in many ways, it's the gold standard elsewhere in the world as well. So it does 3 things, and I've defined them here, but you'll see them on a number of other slides, so don't feel like you have to remember it all here. There's a thing called LOH, which is how many gaps you get on one side of a gene, and it's across the whole genome, and we'll talk about that. There's this NTAI, but it's got a much better acronym called TAI, or just telomeric imbalance, telomeric allelic imbalance, and that is just showing that there are regions of chromosomes missing extending all the way to the telomere. And then large-scale transitions, and all of these add up together to give the GIS or genomic instability score. Now, separate to this, there was the HR Detect test worked out on whole genome sequencing, and this is the classic way we depict whole genome sequencing, which some of you may have heard of and some of you may have had performed on your cancer in the research setting. And this basically aligns all the chromosomes all the way around here in this Circos plot. And then you can see things like amplifications, deletions, and here we have translocations, so abnormal connections between chromosomes that shouldn't be connected. Now, when that is very noisy, and this one is very noisy, that really does look like an ovarian cancer that has HRD, because it's not been able to correct these before they've occurred, and it's resulted in all of these abnormalities. So this is just giving you a bit of an idea about what we're talking about when we talk about damage and the accumulation of damage. And signature 3 is the other way we describe this when we're talking about mutational signatures. So this has more precise information on it, but I'm not going to go through it. You're welcome to have my slides from Ovarian Cancer Australia, and then it's all there. So again, this algorithm just shows you the sorts of things that we think about when we're going to decide if a test is any good. We think it's a biomarker, there should be a phenotype, we should be able to predict a PAPI response, and then we should be able to stratify patients based on what we've learned here and use it in the clinic. So there were germline BRCA mutations originally, then we developed better data for tumour BRCA mutations, which included somatic BRCA mutations. Then we had some other genes, which I'll mention in a moment. Then we had these genomic scars. This GIS score is derived from the SCAR assays, and LOH is a slightly different example. So as a result of all of this, the updated guidelines, which were a year later in 2021, said that all patients with high-grade ovarian cancer should be tested for BRCA1 and 2, both germline and somatic, and patients with the BRCA mutations should receive 2 years of olaparib. But it also then said that testing for genomic instability, or HRD, is recommended because this identifies a subgroup of women who don't have a BRCA BRCA1 or 2 mutation, but who do derive benefit from the PARP inhibitor, and I'll show you some of that data in a sec. So patients with a positive HRD test and a partial or complete response to chemotherapy in the first line, with or without bevacizumab, should receive maintenance first-line treatment with a PARP inhibitor, and that could either be olaparib bevacizumab, according to the PALA-1 trial, or niraparib monotherapy. And patients who were HRD negative did not receive as much of a benefit, and the benefit was not considered to be clinically meaningful, and so therefore those patients were not advised to receive first-line therapy. And we could talk about that a little bit more later. So the HRD tests used in these trials were the Foundation Medicine, which was really mainly restricted to the LOH test, and tumour BRCA. The Myriad My Choice test that I've already mentioned, and My Choice Plus, which just includes a couple of other genes. So I just want to show you this pictorially. So these are the relapse trials, these are the first-line maintenance trials, and these are, just consider these as being major trials. These purple boxes are BRCA 1 and 2. And the further away this box is here from this line, the better. So you can see these pale pink. So these are the ones that weren't BRCA, but were HRD positive. They're not as far away as the dark purple. And this one is mixed with dark purple, so it doesn't count. You've got to go dark purple versus light purple, but it's still a long way away from the line. And the dotted line, which gives an idea of the error, is also a long way away. Now, having said that, here's the wild type. So why am I saying that the value isn't there? So here again, another trial, purple, light pink, but here you can see the green drifting in the first line towards the line. Purple, dark pink, and a bit of drift. Purple, dark pink, and a bit of drift. So I think what's clear is that for HRD, all of these light pink lines are a long way away from the line. So you might then ask, why has this not been approved in Australia in the first line? And one could say it's underway, but to what extent remains a little bit of a mystery. So this is another way of looking at all those clinical trials. And here we can see that the HRD, so BRCA wild type HRD, here again, we're looking at ratios or hazard ratios. And if it's less than 1, it's good. These are all way less than 1. So that is a positive thing. So we will leave that there and now just go and look at some of these implications. I don't have time to go through this in detail, but BRCA 1 and 2 is the key comparator. There are a couple of other genes. Some of you may have heard of these, RAD51C and D or PALB2, occasionally BRIP1. So certainly for these 3 extra genes, we believe that women with an ovarian cancer with those mutations should be on a PARP inhibitor, and we will do our level best to get access to a PARP inhibitor from a company such as AstraZeneca. And then there are a lot of other HRR genes that don't seem to have as much of an impact. The HRD tests do seem to predict for PARP response, and we would like to get access to the testing so that we can broaden the use of PARP inhibitors for women. So early treatment with a PARP inhibitor should be offered to every woman with high-grade serous ovarian cancer as early as possible, unless she develops platinum-resistant disease prior to having a PARP inhibitor. So sometimes in Australia, this HRD test is not done early, and so it is possible that a patient could develop PARP inhibitor platinum resistance at the end of their first line, so within 6 months, because they didn't receive first-line PARP inhibitor, and by the time they have platinum-resistant disease, it is less clear that a PARP inhibitor will help. So PARP inhibitor participation in trials and combination trials should certainly be encouraged, and I'll come back to that at the end. So this is just going to tell us a little bit about HRD tests and the challenge. I've talked about the My Choice test, which is the one we're going to talk about. This is a different test in the lab, which is really difficult and can't be done in the clinic. This tells you a bit about the whole genome sequencing scores that I've already talked about, LOH, which is the one that the Foundation Medicine test uses. And in fact, all of our tests do use LOH, but they use it in combination with these other two tests that I've already mentioned. So this now, is relevant for Australia. So this is the Illumina TSI 500 panel, which we're used to using in things like the MOST screening for most clinical trials. And they are now partnering with Myriad Genetics. So they're going to have the Myriad test involved in the TSI 500 panel. They've also developed a test for large rearrangements in BRCA1/2, which can sometimes be missed, and they've put all of that together. So this now is called this TSI 500 RUO, and it has the DNA variance DNA HRD test, as well as these BRCA large rearrangements. We don't really have that being easily sold in Australia yet, and I think there's still, I'm not clear what the price is, but that is something to watch. Now, the alternative to this, I'll just touch on very briefly, because it's not yet available easily in Australia, but this is called low-pass whole genome sequencing. So I gave you an introduction at the beginning into the concept of whole genome sequencing, because it can basically look with much more detail on a lot of these things. So this is called the Sophia Genetics HRD solution, and Stephen Fox at Peter Mac has been looking into this in a great deal of detail. And we also know that in this, they also do a capture panel that looks for some of the main genes as well, so you're not losing out on that. And this uses a proprietary deep learning algorithm to recognize genomic instability. So it is possible that this could be better than Myriad. There is some data to suggest that might be the case here, where the Sophia test was able to sort out a few false positives that Myriad were otherwise calling as HRD, which weren't. And this is the actual comparison of Myriad versus Sophia. But the broad take home is that there's a lot of agreement here. So if we were to say, which do we want in Australia? Do we want Sophia or do we want Myriad? Well, actually, we now probably do have a choice of either Sophia or the new Illumina combination test. There are some practical considerations, and that comes around what time should we be doing it? Should we be doing it straight early on? And I think we should. But I just want to show you this as my last piece. So this is called the Friends HRD Harmonization Project, and it's run out of the NCI in the US. And you can see a lot of names there of companies that are huge, including Myriad and other companies like Illumina. So what they did was they went and looked at all of these various tests and said, which ones actually work? That this was an in silico analysis. So that means they didn't do it for real, they did it in theory. When they did it in theory, it was a dog's breakfast. They couldn't tell what was good and what was bad. So then they decided to do it for real. So they had freshly extracted FFPE tumor samples from the TCGA. They extracted them at the NCI. They sent them to, I think, 14 different people like Myriad and Illumina and other providers. And the analysis is underway at the NCI, and the data is meant to be out by April. So that is really going to be important, because that's going to really tell us which of the HRD tests is likely to be the most useful. So just to recap, the Myriad My Choice is the reference standard, but the gold standard comes back to these original foci counting tests that aren't practical for the clinic. This HRD RUO test with the add-on to TSO 500 would be practical in Australia because Myriad is not licensed in Australia. We have to send tests overseas for that. And Sophia Genetics would be another alternative. So right now we're awaiting the outcome of the Friends Harmonisation project to tell us which tests might be best. We're awaiting PBAC and MSAC decisions, which I'm pretty sure are probably waiting to see what happens here as well. Once available, I would recommend HRD testing during first-line chemotherapy. We know that for BRCA mutation, we already have PARPI approved for 2 years in the first line until progression in second line, but we actually need to understand better whether patients should stop after 3, 5, 7 years or so. And in the BRCA wild-type HRD positive population, we know that many other countries already approve a Myriad test for treatment in the first line. And if, as a patient, you want to access that, you should talk with your oncologist about whether you can afford to pay for that or access it through research. There are a number of ways of doing that. And then it's 2-3 years of PARP inhibitor in the first line. But basically, we need trials of combined DNA damage response inhibitors, such as PARP inhibitors, with other drugs that are out there to actually make PARP inhibitors to work better, particularly in HRD BRCA wild-type cancers. So they're just some acknowledgements from people in my lab, and just to say that Stephen Fox has been a really big part of helping us understand HRD testing in Australia. And I think I'll leave it on that slide. Thank you.
Thank you very much, Claire, for providing all that valuable information. We just have one question. Someone has said many women are simply not aware of this testing, and this is incredibly frustrating. Why is this the case in Australia?
Basically because it costs $5,000, and until we have the test and a drug approved, it is a very difficult issue as to know whether to raise it. Even if one pays for the test and finds out that a PARP inhibitor would be recommended worldwide, it's also not something normally if someone pays for a drug, you know, you can test after 2 to 3 cycles, after 2 to 3 months, you can do another CAT scan to see if it's working. And then at least you've only paid for 3 months. And then if you've got a response, sometimes you can actually try to get more drug out of the drug company eventually. But this is maintenance therapy. So to pay for 2 years worth, you know, or even 1 year's worth, it costs about $7,000 to $8,000 per month. So it's a lot of money to pay for maintenance therapy. It's hard to fundraise that amount of therapy. So even I, who am very proactive about fundraising, I find it hard to have that entire conversation. But, you know, it's time to be having this discussion. That's why we're having this webinar, and we would appreciate feedback. And there would also be opportunities for women to provide feedback to the PBAC and to MSAC.
And how soon do you think we will get testing in Australia?
I think we need to hear about the Friends Harmonisation project to see if there's a clear winner. If there's a clear winner with an HRD test, then our pathway will be easier. PBAC love clean data. They hate indecision and imprecision. So let's see what happens with that project. And I'm definitely close to the people who will know when the data's through. So we will find out straight away.
Thank you very much, Claire. We really appreciate you taking the time to join us on this webinar.